Next method is the changing asparagine to the other amino acid. So amino acid asparagine and glutamine undergo to the deamidation process. At the higher temperature, it gives the aspartic acid and glutamic acid. Now this obtain aspartic acid and glutamic acid having the unstable and loss of biological activity. So this glutamic acid and aspartic acid is not a thermostable compound. We can understand by using this example triose phosphate isomerase, which is dimeric enzyme, and it is having the subunit, and that subunit having other asparagine residue, and that asparagine residue undergo the deamidation process, and due to mutagenesis the threonine, or we can say isoleucine, is introduced into that. So this threonine or isoleucine is introduced into the asparagine to obtain the thermostable enzyme. Here we are not introducing the asparagine residue with aspartic acid because the unstable enzyme at the low temperature and reduced activity enzyme was obtained if we are adding or we are replacing the asparagine with the aspartic acid. Now next is the residuing the free sulfhydryl group. So this is the third one method to improve the thermostability of protein or enzyme. So the presence of larger number of free sulfhydryl groups lower the activity of protein. In this case, the stability and activity of protein and enzyme can be improved by reducing the number of sulfhydryl group. So ultimately, if any enzyme or protein having high amount of free sulfhydryl group, so its activity is lower. If we are reducing that free sulfhydryl groups, so the activity is improved. Example is human beta interferons. So it is produced in E. coli by genetic engineering, and it is having the 10% antiviral activity, and cysteine residues are there present with the disulfide bond and form the dimers and oligomers. So in this IF and beta having the cysteine residue with disulfide bond, and they are forming dimers and oligomers. Now this IF and beta is present in the E. coli cell, not in the human cell. So to overcome this problem, the cysteine residues is replaced by serine. So we are here replacing the cysteine residues with serine because cysteine having the amino acid and serine having the oxygen. So we are, and both cysteine and serine is similar in activity and similar with the structural activity. So when here we are removing cysteine residues with replacing the cysteine with serine. So ultimately, via this process, we are increase the stability and having the good biological activity of human beta interferons. Next is single amino acid change. This is the fourth one method in which the single amino acid changes is done. The stability and biological activity of recombinant protein is improved by second generation variant, and this is achieved by single amino acid change. So example is alpha 1 antitrypsin. So this is the amino acid which bind and inhibit the action of neutrophil elastase. Elastase is the enzyme who damage the liver, lung tissues, so many normal tissues. So this alpha 1 antitrypsin inhibit the action of that elastase enzyme. Now this enzyme is cleaved into serine and methionine, cleaved, it means opening the bond, opening the structure of a particular substance, particular protein, particular enzyme. So this amino acid cleaved and convert into the serine and methionine, and that free methionine oxidase to the methionine sulfoxide. Now alpha 1 antitrypsin is poor inhibitor of elastase. So we are replacing this methionine with the valine. This valine is similar like methionine, but the new enzyme obtained is used for the treatment of the patient having genetically deficiency of alpha 1 antitrypsins. So here we are replacing methionine with the valine to improve the activity of alpha 1 antitrypsin amino acid. It is a single chain amino acid. Next example of this is insulin. Here the insulin is present as a zinc containing hexamer. Now this single amino acid is introduced into insulin and it improves its stability and also increase the biological activity. Next example is TPA, tissue plasminogen activator. So this is therapeutically used for the lysis of the blood clots causing in the myocardial infarction. So in myocardial infarction it is used as a therapeutic agent. This TPA having the shorter half life about 5 minutes. So we have to giving or administered recurrently. So by adding this single amino acid, we are improving the half-life of TPA. Next and last, fifth method is improving kinetic property of enzyme. So in this, the functional activity of enzyme improve by improving their kinetic property like Km, Michaelis constant, specificity, etc. by the oligonucleotide directed mutagenesis process, and it is required for the enzyme having the industrial and therapeutic benefits. So example is septilisins. So it is a serine protease enzyme secreted by the gram-positive bacteria of Bacillus species and used in industries as an enzyme detergent. Now, the larger scale industrial use is not possible of this substilicin because it inactivate when it come in the contact with the active site, because it containing the methionine which bind close to the active site and oxidize the site. So due to this the activity is lost, and this problem is overcome by replacing that particular methionine with the amino acid. So it is manipulated with the amino acid and used at the larger scale production in industries. Now next is asparginase. So this enzyme is used for controlling leukemia. Leukemia is uncontrollable growth of WBC. In human blood, we are having three kind of cells: WBC, RBC and platelets. So uncontrollable multiplication or growth of WBC known as a blood cancer, or we can say leukemia. So this enzyme is used to control that leukemia. Now IV administration of asparginase, which cleaves the aspargine to aspartate, and this asparginase obtained from the different sources. So it shows the different effectiveness in controlling the leukemia because it having the different property due to its origin. So this asparginase having the low Km is Michaelis constant value, the high affinity for the aspargine and hence more breakdown, and it should be selected to be used therapeutically for control the leukemia. So the asparginase enzyme with having the low Km value is having the higher affinity to aspargine, and ultimately it break down the clot of the WBC or growth of the WBC, so it is more useful. Next is tyrosyl tRNA synthetase. So via using tyrosyl tRNA synthetase enzyme, we catalyze the process, and due to two-step reaction, we get the tyrosine tRNA. Now, application of this protein engineering. So it is used for the cancer treatment study. Next, it is used in the pre-targeted radioimmunotherapy. It is done by this protein engineering. In this pre-targeted radioimmunotherapy, radioactive substance introduced into the to targeted organ, so diseased cell or tissue are affected by this radioactive agent and the normal cells are safe. So this therapy, the radiation toxicity is minimized by targeting the organ. For example, we have to target prostate gland to cure the prostate cancer. So, pre-targeted radioactive agent or radiation is applied on particularly prostate gland, and this is the pre-targeted radioimmune therapy in cancer. Next is to use of novel antibody as anti-cancer agent in treatment of cancer. So in this our body is containing antibodies who fight against the antigen, or we can say foreign material, to protect the body. Here we are developing novel antibody who can bind with the targeted cancerous tissue or cells and cure the disease state. Now, using rDNA technology, mutation, deletion in the protein is done and we get the therapeutic protein. So, in this, we are changing the structure and function of protein according to our need. So, this therapeutic protein used for the cell and gene therapy. By this therapeutic protein, we are producing insulin, interferon, etc. as a biotherapeutic agent, and the protein catonization method is used in drug delivery and development. So these are the application of protein engineering.